Hey everyone, welcome back. We are taking a quick break today for another pro tip. We are staying entirely inside our VS Code notebook and we will learn a pure Python trick that will save you massive amounts of computing time. So here is a very common bioinformatics problem. You ran an experiment and got a list of genes that are mutated in breast cancer. Then you ran another experiment and got a list of genes mutated in lung cancer, and you want to know which genes are mutated in both. Okay, so today I'm going to show you the beginner way to compare lists, and then I'm going to show you how a pro does it instantly using Python sets. So let's jump right in. Okay, so let's define two lists of gene names. So in the real world, these lists might have 20,000 genes or something, right? But we will use a small list for this example. So first of all, we will import a library called time. This is a markdown code. Import time. Okay, then we will create two lists of gene identifiers. So let's call it breast cancer genes and lung cancer genes. Okay, I will copy a particular set of lists from there and we'll use these for breast cancer genes, and we will use this for lung cancer genes. Okay, let's run both. And if I ask a beginner to find the overlapping genes, what they will do is they will immediately reach for a for loop. Okay, and it will look something like this. Let me show you. We will decide a common genes, and after that, they will run a for loop like this. For gene in breast cancer genes, if gene in lung cancer genes, then common genes dot append gene. Okay, very straightforward method. So, and then they can finally print. We'll call it beginner method found common genes. Okay, and let's run this, and this works. Okay, it correctly works. It correctly finds out that TP53 and EGFR are the common genes, but under the hood, this is horribly inefficient because the computer takes the first gene, then it scans the entire second list looking for a match. Then it takes the second gene and scans the entire second list again. Okay, so suppose if you had to take BRCA1, it would scan the entire list over here. And then this will, if you have like 20,000 genes, as I said, your computer just did 400 million operations to find the answers, and it is incredibly slow and time consuming and waste of resource. So what is the probe exactly? So the professional Python coders don't use lists for this. First of all, they use sets. Okay, so this is the methodology that they use. A set is a built-in Python data type that works exactly like a Venn diagram. It is mathematically optimized for comparisons. So to use them, we just wrap our lists in the set function. To find the overlap, which is the middle of the Venn diagram, we literally just use the and symbol, which stands for intersection. Okay, so what we will do is we will use set1 is equal to and convert the breast cancer genes to set 1, and finally set 2 is equal to set and lung cancer genes. What this will do is this will convert both these two things into sets. Okay, and then finally pro common is equal to set 1 and set 2. Simple as that. And finally you can print. We'll call this our pro method found pro common. And look at that, one line of code, set 1 and set 2. Because sets use something called hash tables under the hood, Python does not have to scan the whole list every time. It finds the answer almost instantly, even with millions of data points. But sets have even more superpowers. What if you want to find the genes that are unique to breast cancer and not found in lung cancer. Then what will you do? At that moment, we will use a subtract sign instead of this and sign. So let's see how it is done. We will call this unique to breast set 1 minus set 2. Exactly how we do it in maths, right? Then unique to breast cancer are the following genes, and let's run it. And there you have it, right? Now, what if you want to combine both lists together, but you don't want any duplicates, right? At that moment, we will use the vertical bar, which stands for union. Okay, so all unique genes is equal to set 1 and vertical bar set 2. And then you print it, all unique genes involved, and finally all unique genes. And there you go. And finally, what if you want to find the genes that are mutated in one cancer or the other, but absolutely not in both? Then that moment you use the caret symbol, which is the symmetric difference. Okay, so what we will do is we will call this exclusive genes, and what we will write is set 1 caret symbol set 2, and we will print this similarly mutated in one but not both, right? And then exclusive genes. And there you have it. And that is your pro tip of the day. And next time you need to compare two lists of genes, patients or k-mers, just stop writing for loops. Convert them into sets and use basic math symbols to generate your Venn diagram instantly. I hope this saves your computer some serious processing time. Drop a like if you learned a new Python trick today. Make sure you are subscribed to our channel, and I'll see you in the next video.